Copyright University of Auckland, all rights reserved. The content and delivery of lectures in this course are protected by copyright. Material belonging to others may have been used in these lectures and copied by and solely for the educational purposes of the university under license. You may record the lectures for the purposes of private study or research, but you may not sell, alter, or further reproduce or distribute any part of these lectures to any other person. Failure to comply with the terms of this warning may expose you to legal action for copyright infringement by the copyright owner and/or disciplinary action by the university.
Kia ora tātou. Nau mai ki tēnei rā motu hākea mō te Liggins Institute mea te whare wananga o Tamaki Makaurau. Good evening and welcome. My name is Frank Bloomfield and I'm Director of the Liggins Institute, and it's my pleasure to invite you here this evening to the Liggins Institute lecture series for 2017. This evening's lecture is The Power and the Potential of the Gut Microbiome, otherwise known as gut bugs. And I hope that both you and your personal complement of gut bugs have been able to enjoy the refreshments before we get started. So before we do start, just a few housekeeping notices. Bathrooms are either directly across the atrium outside or along the corridor to the right as you leave the lecture theatre. And if a fire alarm should go off, there will be flashing red lights and a very loud noise. And if you could all exit through either the two doors at the back or the emergency exits down the front here and congregate outside the building in the large space out the front there. Finally, if you have your mobile phones with you, please turn them to silent or off. Thank you. So we do have a film crew here this evening who are making a documentary about the gut bugs trial. And they'll be filming a small group of people who have given their consent to be filmed. But we'd like to reassure you that they will not be filming anyone else, and no one else in the audience will be recognizable on the filming for the documentary. Also, some of the speakers this evening will be sharing their personal stories, and we'd ask you not to take any photos or any video or audio recordings of the proceedings tonight. Thank you. So I think we'll get underway, and our first speaker this evening is Dr. Justin O'Sullivan, who is the Associate Director for Research of the Liggins Institute. He is a molecular microbiologist and therefore a key member of the team of the gut bugs trial as he understands what's going on with those microorganisms in our guts. Justin took a first-class honours degree in cellular molecular biology from the University of Canterbury and then a PhD from the University of Otago, followed by a training in Oxford before he returned to Auckland, first at Massey University and more recently here at the University of Auckland. So Justin, if you'd like to set proceedings off for us.
Thanks very much for coming and listening to us. I got told that there'd be a small audience. Someone told me porkies. So today what I want to do is take you on a bit of a journey. And the journey I'm going to take you on today, hang on, I'll get this to work. All right. Journey I'm going to take you on today is basically onto your insides. So of course, this here is a picture of a colonoscopy going into somebody's bowel. And it looks really lovely, really clean. Obviously they've had preparation which has made sure it's empty. But as you see and you're looking at this, you know, this is a really interesting environment, okay? It's the largest endocrine organ that you have in your body, and it's very, very important for your health and your well-being. But it's not just the stomach or the tissue that we're looking at that's important. It's what lives there, okay? And basically, your guts contain trillions of microbes, literally trillions of microbes. And these microbes are all different. So what we're looking at on this particular slide are cartoons of them because it's a bit easier to show. But there are things like this, and some of you may have been unfortunate enough to have had this little beastie here. This is Giardia, okay? So if you've been camping or tramping and you've drunk water from the still part of the river instead of from the fast-flowing part, you come away a few days later, you have a very bad case of diarrhea. It's because of this little beast. possibly. And that's not a really good one to get. This here looks like a moon lander. In fact, the understanding is that the moon landers were designed on the basis of these little beasts. This is a phage, and this is for bacteria, so it's a virus for bacteria. Billions of those in your guts. Here we have just representations of some of the bacteria that you can find in your stomachs, these spirochetes and cocci and some rods here. And these all happily live inside your guts, but they also live on your skin, everywhere basically, except I think on the iris of the eye. So how do we know this? Yeah, so we don't know it because of this, right? This doesn't work. Because bacteria, when you look at them down a microscope, they look the same. There might be cocci, there might be bendy, twisty things, there might be rods. But really, they look the same. They don't have faces and things like we recognize. So in order to look at them, I'll show you a very simple cartoon. The way that we do it is like this. We have a population of bacteria. In this instance, they're little robots. Okay? Now, looking at these, we can tell they're apart, or tell them apart just by looking at them, because they have this little blue and yellow and green barcode on them. Okay? Bacteria, same deal. What we do is we take them, we chuck them in a blender, we break them down into their component parts. Cool. But it's still pretty hard to tell what they are. So what we do is we photograph them, and we do this using molecular methods. We do this, and in fact what we're doing is we're photographing part of the DNA in the bacteria. And when we do that, we're able to effectively Xerox it and amplify up the sequence that we're looking at. And by doing that, we can then sequence that. So we can take the photograph, if you will, and we put it through the computer. And when we put it into the computer, we do a very simple thing. We have fragments of the bacteria, fragments of their DNA. And these fragments of their DNA represent a really critical enzyme in bacteria, in humans, and every organism that we know of practically except viruses. Okay, and the part that we're looking at is this factory, a factory that makes proteins. And it's really highly conserved between all of the organisms that walk, talk, bacteria, fungi, etc. The cool thing about that enzyme is that there are parts of it that are different. And those parts of it are, in fact, these little barcodes that we see here. Now, in these robots, when we take them in and we do this, the photographed images of them, of this barcode, when we put it in the computer, we effectively just do a matching. So we use a matching program. And it says, does this equal this barcode on this robot? No. So do the screen again. Does it equal the barcode on this robot? Answer is yes. And so therefore, the identity of the robot that we smashed is, in fact, this one. And it works just like that for the robots that I've shown you here, for the bacteria, and for the fungi that are in or on your bodies. Nice. So that's how we do it. Now, we like to think that we're very special. We like to think that we're all unique. And we like to think that's because of our DNA. And to a degree, you're right, it is. We all have about 3 billion base pairs of DNA in our bodies, or in each one of our cells, sorry. And it's all, between each of us, there are a whole lot of variations. In fact, about 1 million differences between me and anybody in this room. But if I was to take somebody and blend them like that robot and extract all of their DNA out of them, okay? I'm not allowed to do this, by the way. But if I was to do that and then take all the DNA and line it up end to end, then the DNA you have in your body, if I took from all 10 million cells that you have, all right, 100 trillion, all right, but only 5% of them actually carry DNA. I'll tell you about that in a second. But if I did that, okay, your DNA would go from the Earth to the Sun and back 66 times. Okay? That's how much DNA you have. But it's all repeats. Like, it's one copy and another copy of the same thing and another copy of the same thing and another copy. You know, it's actually about 5 trillion copies of the same stuff. And it would go from here to the Sun and back. It's a lot of times. If I took your bacteria, all the bacteria that live on you, which is about 100 trillion bacteria, okay, live on and live in you. If you took them all out of a body and you put them on the scales, it'd be an organ about the size of your liver, okay? That's how much bacteria there is, right? Bear in mind that you can't see bacteria except under a microscope. So about the size of your liver. That's a lot. If I took the DNA out of them, how far would it go? Unfortunately, it wouldn't go as far as your own DNA because bacteria are much smaller. Their genomes are a lot smaller. In fact, their genomes are about 1,000th or 2,000th the size of yours. Quite small. So their DNA, if I extracted them out, would only go from Earth to Mars. Mercury. Yeah, not a astronomer. Yeah. All right, Mercury. That's kind of helpful. Thanks. So over to Mercury. All right, once. That's it. But that's a bit misleading. And it's misleading not just because it looks like it's a lot less, right? But it's misleading because it comes from 100 trillion organisms. Now, in your gut, there are about 1,500 different bacteria, okay, at least. They all have different genes. So you and I have about 24,000 genes in our DNA. And that 24,000 genes contribute to make us partly who we are. Flies, some flies have 6,000 genes, 20,000 genes. Some amoeba have more genes than you do. Common weeds have more genes than you do. Bacteria don't. They have a lot less. They have about 4,000 genes each. But if you take all the bacteria on your body or in your guts and you look at the genes they have, then in fact what you see is this. There are over 1 million microbial genes in or on you, and you have 23,000. That's kind of skewed, right? So these organisms are really important. They have a huge genetic component, a large coding capacity that enables them to do things that you can't. It enables them to provide you with functions that you don't have encoded in your own DNA. And this is really, really important. All right? Because for a long time, ever since a guy called Koch, we've looked at things in this type of way, which is a them and us situation. And we've labeled bacteria as a them because bacteria, things like cholera, anthrax, leprosy, right? The organisms that cause those disorders, they kill you. That's pretty simple. So it's a them because we should be afraid of them. But that's not the case. Because a lot of these bacteria, the ones that live on you, that live in you, they're commensals. They work with you. They help you develop. They help your gut develop, okay, as you grow. They change your mood and various other things that Wayne's going to talk to you about in a few more minutes. However, having said that, there are some that don't. This is a pseudomembranous colitis, and this is caused by overgrowth of this organism called Clostridium difficile. All right? If you happen to get this, it's very unpleasant. Okay? So you'll have very bad diarrhea. A lot of these are actually resistant to antibiotic treatment, and that's not a good thing. So, overgrowth of certain organisms can be bad, but your microbes aren't all bad. So a lot of them actually help you eat, grow, and be healthy. All right? So some of them will take products like milk, and they will happily convert it into something else that we like to eat. They'll do this before we get it, or they'll do it when it's in your stomach or in your intestine. Some of them, of course, will make it go sour, and it's just not quite so nice, but it's a balance. Some of them, all right, they'll make new products, things that change the way you behave, things that change the metabolism in your liver. They might make short-chain fatty acids, for example. They might make other products, steroid precursors, various things like this that actually change the way you feel and you behave. And in so doing, you know, you also get these ones here. They make these novel things. Some of them, they'll modify foods. Some like this one here, in this instance, just take a wee bit of the ice cream. Other ones will take it all. Okay? But they do these things, all right? They modify the things. And so effectively what we're talking about is a very complex relationship. And that complex relationship moves and it grows over time. So your microbes are something that you are born with. And as you go through life, so this here is just showing the diversity, so the types and the numbers of bacteria, right? So it's increasing. So as you go from your very birth at time 0 out through 2 years of age, the number of bacteria you have in your gut increases and goes out like this. If you happen to develop type 1 diabetes during this time, you see that this changes. So the number of bacteria you have is much less, the diversity is much less than what it would be if in fact you went to a normal individual. And this is because they're linked, because your physiology is linked to the bacteria. You select for some of these bacteria, as well as they inform you and your development. So your microbes, they develop over time, but they respond to nutrition. You can change them with what you eat. Okay? And this is shown here very, very nicely in this graph here. And these are different populations around the world, and they all have very, very different diets. The population in yellow up the top here, the Hasda, are hunter-gatherers. And these hunter-gatherers have quite a different population. You can see that because they're all clustering up here, right? This yellow group. Much, much different from the Italians down here in this light blue color, right? They're a long way away. Or from the people down here in brown who are the Burkina Faso, who are from Africa. Okay? And so you see that the diets, the people, the way they live, change the microbiome. All right? And it correlates with that. But over the course of your life, and this is work that was done by one of our team, Fellini, who's here somewhere. And Fellini did this work with Billy Apple recently. And Billy did this crazy thing where he kept some toilet paper from 46 years ago. He's an artist, right? So it's good. And so he kept this material for 46 years, dry, stored, perfect, right? And then he came to us one day and he said, "Can you do my microbiome?" We said, "Sure." So he gave us some fresh samples, and he gave us the old samples. And what Fellini did was she looked at how the microbiome differed between these samples over 46 years in this guy's life. Now, when he gave the first samples, he lived in America. When he gave the new ones, he lived here. He's now 80, he was 40. All these things changed, right? All these things changed. But his microbiome, part of it was the same. A big part of it, about 50% of his microbiome was the same 46 years after. And this is his dot down here. And it follows a predicted curve, right? Really, really well. And that's 46 years later, it was still the same. A large component of it. And the reason for that is that you are unique because of your DNA. And part of your DNA actually selects for some of the bacteria that live in your guts or in and on you. So that's pretty cool. So just like your genome, okay, it's here. This is the environment, this is the trash, all right? So just like your genome, you inherit your microbes, all right? You inherit them from your mum. And your genome, it selects for some of them and throws some of them away. And the environment gives you other ones, okay? And so it changes over time. But there's a core component that stays the same throughout your life. And this is really important. But the key thing about this is that you do inherit your microbes. Sorry, I'm going a bit late. So you do inherit your microbes. In fact, you inherit them when you're in the womb. So before you're born, when you're developing in your mother's womb, you inherit microbes. And that's what's shown very clearly here. This is the meconium from an infant here. And you see that it's different to anything, any pattern from the mum. It's very, very specific to the meconium, not the skin or any of the other sample sites on the infant. So you inherit this in the womb before you're born. All right. And the key thing about this is that just like this spaceman, microbes are specialized to transfer from host to host. And this is really important. They transfer from host to host. This is what they do. This is how they move. So most of them wear a little spacesuit like this, okay? And that enables them to transfer from host to host. Now, I do have a video I was going to show you. I'm running out of time, but I'm going to do it anyway. Oh, no, I'm not. Yeah. Hang on. I have to escape out, sorry. It didn't work very well. All right, hang on. Here we go. So the way they transfer, right? Have you ever seen this? Yeah, this is great video. It works really well with the sound, right? But clearly, okay, this is one form of transfer that happens. I'll speed it up a little bit because we don't have much time. Right, but you can tell what's happening, right? And so just, I'll let it run. Just in a second, you're going to see a specialised form of transfer, right? This is for viruses, all right? Bacteria don't do this, not so much, but they are present there. All right, it's coming. Yeah, just got it. That's the build-up that's important, right? So it's coming, and we're about to get the snotsicles, and so it very, very specialised form of transfer, this one. All right, this is why you should always cover your mouth when you sneeze. All right, so it's about to come. Here it is. So the floodgates open, and the viruses basically spread, you know. Okay, and they go a long way. That's lovely. Okay, so you're seeing transfer right here, right? This is why you should always, you know, enter your elbow, because you will transfer your microbes. All right, and so I'll cut that out now. Okay, but what I want to do just quickly is one more point. So that microbes are specialised for transfer, right? But, you know, effectively, just as I hand over to Wayne, I'll show you something different, okay? Because, I've got too much on my hand. Sorry. Because transfer happens all the time, okay? So my hand's clean, you know, now, relatively. All right, and as Wayne comes in, and I'll hand over to him, you see, so we shake hands, you know, and that's cool. But now I'll show you what happened when we did that, okay? It wasn't just that we had an awkward moment shaking hands. It was, in fact, that what happened, if I go, I think it was this one, and then you still got a hand, hang on, I'll go to the black yet. If I do this, now, hopefully you can see, because I don't know if you'll be able to see, but, because the lights aren't too dark, but my hand is glowing, yeah? And it glows because what I put on it is actually this powder. Well, it's a liquid. And when I shook Wayne's hand, then his now glows as well. Okay. And in fact, if you do this with a row of people, you can pass it just from one hand like that down 5 people, and you can clearly see when people shake hands one to the next, it'll move from one to the other. And I've done it with kids, and it goes down about five people. Kids are easier to use than, you know, because they like to participate. So that's what happens, okay? And this is something that we're trying to use. This is what we're trying to use. We're trying to use this natural tendency, this evolution of these microbes that help you develop. They're important for your health and well-being. We're trying to use that, and we're trying to use their natural characteristics by putting them into capsules and transferring them from individual to individual. And with that, I'll pass over to Wayne. Thank you.
Thank you very much, Justin. So there'll be time for questions at the end once all the speakers have finished. So our next speaker this evening is Professor Wayne Cutfield. Wayne is a paediatric endocrinologist who works clinically at Starship Hospital, as well as running a large research group here at the Liggins Institute. Wayne got both his MBCHB and his MD here at the University of Auckland, also trained in Cincinnati, and he's a previous director of the Liggins Institute. Currently, Wayne is director of one of the National Science Challenges of New Zealand called A Better Start. Wayne, thank you.
I have to keep pulling my pants up because I've got so many microphones on, they're pulling them off, so you'll have to excuse that problem. Thanks, Frank. Good evening, ladies and gentlemen, and fellow gourmands. You've heard from the sartorial intelligence of Justin, and now, as my team would say, you get to hear from the crusty old demon. That's the way it is. Now, we are in the midst of eating our way into an obesity pandemic that encapsulates the entire world. The World Health Organization in recent years declared that obesity and related diseases is the biggest health problem the world faces. Ironically, it replaced malnutrition and undernutrition as the biggest problem the world faced. And in New Zealand, we're not faring so well because on the OECD obesity charts, we come in number 2 as the second most obese nation in the OECD of the 36 member countries. We're ahead of the US, and we're well ahead of the UK and Australia. So we're not faring so well. Now, I need your participation for the next few minutes. And you're thinking, he's not collecting stool samples from me. I promise I'm not. And I'm not going to give you stool samples. But what I need you to do is I need some of you to stand up. And Fellini, go back one more row, Fellini. So everybody in those rows forward, please stand up. Okay. So as you're standing up, those of you who are standing can see there are a lot of you standing. And those sitting are looking at you, and they see a lot of you standing. Think for a moment. This is your first day at school, your new entrance. From New Zealand data that's currently under review that we've analysed, and it's one of the biggest studies of its kind in the world, 400,000 children aged between 4 and 5. This is the portion of you, when you start school, that are overweight or obese. 1/3 of you, 1 in 3. Back you go, Fellini. Stay standing, please, just for a moment. Can those stand up who are in line with Fellini on both sides? Up we come. So it's even more. It's a lot more. So this is the proportion of you who are now Pacifica new entrant students. More than half of you are overweight or obese as you start school. Or if you are from a less affluent family, this is the proportion of you who will be overweight or obese when you start school. Now, can I have that middle group in the back row sit down? Why did I make you sit down? Treatment of established obesity in childhood is somewhat ineffective. If you look at long-term trials using diet, activity, education, or even drugs, and drugs are generally not licensed to be used in children for weight management, but if you look at those, the combined effect, that's optimistically the size of the effect those interventions have. So you can see, and you can all sit down now, so thank you, and it wasn't as bad as you thought it might be. You can see that this is a major problem, and we need to look at effective ways when obesity is established to finding solutions. Now, I thought this was an interesting slide. It's got a few interesting comments on it, because now I want to get into the gut microbiome. Now, you've heard from Justin about how many bacterial species we have, but there are a couple of other interesting things. For those of you, how many of you hop on the bathroom scales more than once a week? Go on, be honest. Go on. It's more of you than that. Okay. The good news is that you are between 1 and 2 kilos lighter than the scales tell you, because they're your gut bugs, and they weigh as much as your brain. And you cannot say you know what for brains. But I thought the last comment from Michael Spector was interesting. And he's talking about the gut microbiome. They play such a crucial role in our lives that scientists have begun to reconsider what it means to be human. Now, what we know about the gut microbiome and its role in obesity and metabolism is pretty new. And in the last 10 years, this topic has exploded and is probably one of the hottest topics in medical research. And the story begins with this little fellow, the germ-free mouse. Now, this mouse lives in these sterile component cages, and they have no bacteria in their gut. And one of the first studies by Fred Backhead and colleagues, what he and his colleagues did was they took the gut bacteria from normal mice and put them inside the germ-free mouse. And they put the germ-free mouse on a diet as well. And the germ-free mouse got fatter, appreciably fatter, and had a greater risk of developing diabetes. And then suddenly it became clear that perhaps gut bacteria might influence our weight and our risk of obesity. Now, one of the interesting things about mice that are different to us, well, two things that are different, which is great for studies like this. They eat each other's poos, and we don't do that very often. And the second thing is that the pH of their stomach is much, much less acidic than ours, and so when they eat those bacteria, they're not going to get munched up by all of the acid in their guts. So they've been an incredibly important model to study. So extending that further, and these are the germ-free mice. The germ-free mouse adopts the physical characteristics, the phenotype, the shape of the donor. So you can see on your left, it's a normal donor mouse to the germ-free mouse, it stays the same size. If you take a fat mouse to the germ-free mouse, it gets fat. But if you take a very thin mouse to the germ-free mouse, it gets skinny. And that just adds to the importance that the gut microbiome has in these early studies. Now, while these studies were going on, studies began in humans, and they weren't intervention studies like I've just shown you. What many researchers have done, and there are now hundreds, thousands of publications, many thousands of publications, and what researchers have done is they've looked at a condition and then said, is the gut microbiome different, or are there different aspects about it in that condition? We call that association studies. And these association studies show that there is less diversity of the bacteria in those with obesity, and we believe that diversity is really important. Think about a rainforest versus a pine forest. It's much more resilient than a pine forest. The gut microbiome is different in those with type 2 diabetes and those with allergic diseases like asthma, eczema, and food allergies. It's also associated with inflammatory bowel disease, but it goes further than that. The gut microbiome is different in a whole lot of neurological and behavioural conditions, such as depression, autism, Parkinson's disease, Alzheimer's disease, and mania. And so you could speculate from these human studies that the gut microbiome has extraordinary effects on us, that it doesn't just affect our weight, it affects our risks of diabetes and bowel disease and allergic diseases, and it affects our risk of mental health diseases, and it affects our behaviour. When you next go to, and in fact there's data that suggests it influences our appetite. So when you next go to muffin break and have that muffin, who wanted the muffin? You or your gut bugs? But I'm telling you about association studies, and association studies don't prove cause and effect, as that cartoon says. Diabetes has increased dramatically over the past 20 years. That proves that diabetes is caused by global warming. Now, we know that's not true. If two events occur that occur in parallel, that they're not necessarily related. It's not cause and effect. And so for that, we need to do more studies. Now, we're moving away from the germ-free mouse, because he's slightly odd and he's got no gut bugs. And we all have lots of gut bugs, as Justin said to you. And what has been shown is that if you take a slim mouse, provide the gut bugs to a very fat mouse, then that fat mouse will lose a third of its body weight. It's a pretty dramatic effect. But you can reverse that same process by taking the gut bugs from a fat mouse, give it to the slim mouse, and that mouse then gets fat. Now, let me tell you very quickly about a whole bunch of studies where these are intervention studies where you take the bacteria and put them in another mouse to see what effects it has. So I've talked about the slim mouse, obese mouse, but they've also mixed humans and mice up and used the human as the donor, not the recipient. Mouse to human doesn't go down well. So if you take twins and you take the fatter twin and the slim twin, and if you put the bacteria from the slim twin into a germ-free mouse, it stays slim. And if you put the bacteria from an obese human twin into a germ-free mouse, it becomes obese. And then if you mix these two up, here, and you go from here to here, the obese mouse gets slim. So you can just keep manoeuvring the phenotype around by transferring the gut bacteria. Interestingly, and this just shows you the potential ramifications of our gut microbiome, if you take anxious humans who have irritable bowel, and that's a lot of us, including me, and stick it in a germ-free mouse, the germ-free mouse gets anxious and develops irritable bowel syndrome. Okay, that's mice and humans to mice. What about human to human study? And most of the work that's been done is in this awful infection that Justin alluded to, chronic Clostridium difficile infection, a life-threatening infection with awful diarrhea and has a high mortality. Now, what they've done, and there are a number of studies showing it, they take usually a family relative who provides a stool, they kind of clean it up, put it in a blender, make a smoothie, pour it in a tube. It either goes down in a tube down that way or in a tube up that way, and look at the dramatic effects. between 80 and 95% of this potentially life-threatening condition is cured. The treatment on the right, vancomycin, is much, much less effective, which was conventional treatment. And so that is now becoming a common first-line treatment for this awful infection. Now, what about other human studies? And there are two others. I told you about this awful Clostridium difficile infection. So there was a woman who was treated, and the donor was her 16-year-old daughter, who was overweight and rapidly putting on weight. So the bacteria went from the daughter to the mother who had the infection. She was cured, and guess what happened to her? She became obese. So there's an accidental situation in which the reverse has occurred with what we're trying to seek. And since then, they have more rigorous screening of donors. You need to know more about the donors because they are your treatment. Now, there's a tiny study of 9 individuals who had diabetes, and they donated gut bacteria from those who didn't have diabetes, and their diabetes improved. So it was a very tiny study. It was a very short-term study. So that's as much as we know in human studies. So not a lot. How then do gut bacteria influence our weight? And they do it in multiple ways, not just in one way. They increase the number of calories you absorb by about 200 a day, which is a muffin and a half. And they do that by breaking down waste that you can absorb. So non-absorbable waste is broken down into components like short-chain fatty acids you can absorb and then use as energy. They actually accelerate and promote fat synthesis and storage. They also promote inflammation, and they do that and that leads to increased fat synthesis, but it also increases the risk of diabetes through these pro-inflammatory mechanisms. And as I mentioned, from animal studies, it appears they affect appetite. They may increase or decrease your appetite. So they seem to have effects in lots of places, and it's not all just sitting in your gut. Some of these effects are based on products that get into your blood circulation and get all the way to your brain. So now to our study, the gut bugs trial. About 18 months ago, close to 2 years ago, Justin and I have offices beside each other. So we're kind of in each other's rooms like yo-yos. And we sometimes annoy each other, but we have good ideas and we bounce them off each other. Some of them are good, some of them are just plain silly. And I said to Justin about 18 months, 2 years ago, we need to just do a treatment trial. There's all of this association studies. It's time that we move forward, and we did a treatment study. And so we decided that's what we'd do. And the interest shown has been enormous. We've barely started the study. So we've got a cover story on the listener. We were on the project on TV3, and Razor are really interested in making a documentary series about the research. So clearly it's an area of great interest, and that's why you're all here. Here our team, with all these smiley faces. It's a big team. And the reason it's a big team is because they've got huge range of skills, and that's what we need for a study like that. And I think we've got one of the strongest teams that could do research like that anywhere. And there are teams around the world doing similar kind of research. We've got clinicians, we've got endocrinologists, project managers, clinical scientists, we've got molecular microbiologists, we've got research nurses, we've got nutritionists, Pacifica researchers, and we have bioinformaticians. And you think, well, what bioinformaticians, why do we need those? And the reason we need William is because this study will generate more than 2 billion pieces of information to analyse relating to the clinical things we measure and all of the gut bugs. 2 billion pieces of information, it's huge. The last person on the list is important, and that's Tim Edney, who is our philanthropic sponsor, and we will hear some of his comments from Frank later. He's in Arrowtown at the moment and couldn't be here, sadly. And I also want to mention the Liggins Institute Trust, and also a Better Start National Science Challenge, because they gave us the initial money, and Tim has become our major sponsor, which has been great. And he's also part of the team. He comes to the weekly meetings, and he's in there, boots and all. Now, in studies like this, the donors are critical because they're providing you with the treatment. And what's really interesting in other studies like this, they don't make much effort to characterise the donor or even understand what are in their bacteria. So who are our donors? And on the project, they made a comment that it's harder to be a donor in the study than it is to get into Harvard, Now, that's not quite true, but it is pretty tough. Our donors are slim. They have measured low body fat, and we measure it. They have to do a lot of exercise. They have a healthy diet. They don't have any health problems. They're on no medications. So they're a very special group. To get our 4 female donors, we started off with 65 eligible donors to get down to our 4, because that's how important it is to have the donors and their bacteria right. So here's the study design, and I've tried to make it as simple as possible. I can't share results with you. We're kind of kicking off, and we're not getting far yet. Well, sorry, we are making rapid progress, but we haven't got very far yet. I've talked about our donors. Each recipient is exposed to 4 donors, and that's to create that variability. No other study has multiple donors for one recipient. What we have developed from our donors is a novel way to give the gut bacteria, because you've seen the pictures of the tubes, right? The tubes down and the tubes up. How many people are going to say, pick me for a study like that? And we're talking about teenagers who are 14 to 18 years old. So this is where Justin and Darren have been hugely helpful, and that is developing a method to take a free stool, clean it up, get the bacteria out, clean them up, put them inside one capsule, put them inside another capsule, more complicated than handmade chocolates, cleaner than handmade chocolates, and you swallow them. And then they are broken down and released in the bowel where they need to be. And we've shown that very few bacteria are lost during that initial process and exposure to air, and we can put them in the freezer and then thaw them after 1 week, 1 month, 2 months, right out to 6 months, and they're still fine. When we thaw them, they're still fine. So that has been hugely helpful in terms of how we're going to treat our recipients. So we've got 40 females, 40 males. Our participants, to be eligible, need to clearly have a weight problem, and we've clearly defined that. And we randomise them. We randomise them to those who get treatment with the gut bugs and saline. We don't know who's getting what. The recipients don't know what they're getting. Before we do that, we want to give the bacteria the best shot, so we clean their bowels out, and we give them a low dose of bowel preparation to sort of partially clean their bowel out, so these donated bacteria got their best chance at getting established. And then they take their capsules, and they take 28. They have 16 one day, 12 the next day. And it's one treatment, that's it, one treatment. And we follow them up at 6 weeks, 3 months, and 6 months. And what we hope to see is weight loss, and we'll wait and see. Having said that, we don't just measure weight. That's one outcome, and it's the most important outcome. But we want to know how much fat there is in their body and where that fat is distributed with treatment. We want to know what their diabetes risk is and how well insulin works, and we're going to study that very intricately. We want to look at their blood pressure, not just once. We're going to measure it right round the clock every 20 minutes to see what the blood pressure profiles are. We want to look at how well the liver's behaving, lipid levels. We're going to characterise all of those bacteria in the recipients and plot how they change, those trillions of bacteria. And we're going to look not just at the bacteria, but the things that they release that get into your bloodstreams and my bloodstream, and measure those. And we're also going to look at their psychological well-being, because there's been so much made from association studies about gut bacteria influencing our behaviour. And that could turn out to be incredibly important. So we are measuring many, many things in our recipients. I have to say, what is really amazing about our study is that our donors are amazing people. They give up their time. It's incredibly inconvenient. It's very uncomfortable. And when you ask them why are they donors, they want to make a difference. They want to be part of something that they think is important. And equally, our recipients will shatter any expectation you have of who our recipients would be. They're extraordinary people. They're incredibly smart. They've come to us. They understand what the study's about. They've tried lots of options that haven't worked, and they too want to be involved in something that makes a difference for them and all of their colleagues. And that's been the most impressive thing so far in the study that I've noticed. So thank you, and I think I'd probably have a cheeseburger myself.
you very much, Wayne. So our next speaker is Dr. Ben Albert, who is a postdoctoral research fellow at the Liggins Institute, having recently obtained his PhD from research done at the Institute and also recently obtained his final qualification to be a paediatric endocrinologist. So Ben took his medical degree at the University of Auckland as well, and he is now supported by a fellowship from the Maurice and Phyllis Peichel Trust that is supporting him in doing research at the Institute. Ben is very involved in the hands-on end of the gut bugs trial, we might say, interacting with the participants, and he's going to tell us a little bit about that now and introduce some of the participants to us. Thank you, Ben.
Cool. Thank you, Frank. As I talk around this. Yeah, cool. Oh, I think I'm good. It's really great to get to talk today, so thank you very much. I was asked to join this study to be one of the people that translates it into an actual study with people coming into the Institute, having tests, having treatment, and being assessed, which, from my perspective, is the most interesting part, because the people who are volunteering to be part of the study are really fantastic people. And I was really excited to be part of a study like this because, as Wayne has demonstrated to you, obesity is one of the world's greatest health problems. It's an increasing problem in young people, and our treatments don't work. So we need effective, innovative treatments, and this is a really exciting new approach to obesity. So as part of recruiting people, bringing them through the trial, it's been my job to explain the study to them, to answer their questions, and then take them through the assessments and treatments as they happen. And one of the things I wanted to tell people is that being a participant in a clinical trial is not a minor thing to do. It involves contacting people you don't know to answer an ad that came to you out of the blue. It means talking to nosy people like me who ask really awkward questions about your health and your life. You have to come to a research institute, which is really shiny and nice, but quite scary as well. You have to undergo assessments and tests and take an experimental treatment. And the thing about taking an experimental treatment is you don't actually know what effect it will have on your body. You hope that it is going to have positive effects, but you don't know. If we already knew that it worked, then it wouldn't be an experimental treatment. So the people who volunteer for a trial like this are slightly unusual just because they were excited and interested enough to come and be part of a trial. But they're also really brave and generous people who have hope for themselves, but are also trying to help the other people who never came to join the trial. The participants who have volunteered in this trial are particularly impressive people. They're really intelligent, enthusiastic, thoughtful young women. I say young women because at this stage of the trial, we've only had women come through. They're worried about their weight and their health. They've thought about diet and exercise, and they've made efforts to try to improve their weight. And some of them have been successful, but only briefly, and others have been unsuccessful. And that is the common experience of people with a weight problem. It's really hard to change your weight. And these people really carefully read all of the information we provided them, more carefully than I did. They quizzed me about their information. They'd done their own reading before I called them. Really, really impressive. And I think I'd really like you to go away understanding that the stereotypes that we might have about people with weight problems and young people with weight problems are very frequently wrong. One of the participants in the trial, Alofa, has offered to come and speak to you a little bit today. She's going to tell us a little bit about herself and why she wanted to be part of the trial. You can come up now, Alofa.
Hi all. As you know, my name is Lofa. I'm 18 years old and I'm Samoan. I'm the eldest of 5 children and 19 grandchildren. I was previously head girl at Henderson High School out west, as well as youth MP for Awharingaro, at the 2016 Youth Parliament, and this year I'm currently involved in the steering committee for the New Zealand and US Embassy Youth Council. I'm studying a double major at Auckland University in ancient history and mathematics. In my spare time, I enjoy reading and watching horror movies, thrillers, and action. I love to play sport. I love to play hockey, league, netball, boxing, anything with physical contact. But if anything, the most important thing you should know about me is that I'm a participant in the gut bugs trial. Now, why am I doing this? As you can see, I'm not exactly the ideal size for an 18-year-old, and I believe that this experiment can help me with my weight. I wouldn't say that I've struggled with my weight because I've never been fussed about it. Yes, there have been times where I couldn't do certain activities, but then again, being the size that I am meant that I was always picked for first 11 on the field. I've tried losing weight through natural processes such as drinking more water, eating the correct meals at the correct times, exercising and weight loss challenges, and also some not so natural means such as the Garcinia Cambogia pills. And as you can see, these haven't really affected my body in the way that I've hoped. Losing weight to me would mean that I would be able to do recreational activities that everyone else can do, such as the flying fox at camp, and not having to spend hundreds on a dress plus shipping because Kmart didn't have my size. The second reason as to why I'm doing this is because I truly believe that this trial will bring groundbreaking results that will help those in the future who struggle with their weight. As a mathematician, I am in awe and humbled at the opportunity to be a part of something where the science just blows me away. I'm excited to be a part of something revolutionary, and if it doesn't work, then I'm glad to have been a part of the testing process in which the pills could be further perfected. I just want to say thank you to the team for being hospitable and for doing everything that they could to make this journey as comfortable as possible. And I'm excited to see the end results, and I hope that you guys are too. Thank you, and enjoy the rest of your night.
Thank you very much, Alofa. Obviously, I don't need to emphasize anymore just how impressive Alofa is to come up and talk today, how insightfully she talks about the problem that she's had, the things that she's done to try to improve it, and the other participants in this trial are equally impressive as Alofa. The other people I want to talk to you about in the study are the donors, and you've heard about the donors. They're really fit people who exercise is a really important part of their life. They have a healthy diet. Actually, many of them are exercising at an elite level. And what's really special about them is there's not a lot in it for them. Usually when you come in to be a trial, you're doing it because you're hoping to get some benefit for yourself. And the donors in this trial have really got no benefit for themselves at all. They've entirely done this because they're altruistic and want to help others. And in fact, when we advertised for donors, we put up an ad that said, "Would you like to share your good health with others?" And we wondered how well that would work. And we were instantly flooded by 70 people who wanted to be donors in the study. So there is also this great resource of people who really, really want to help. One of them said to me, "Well, who wouldn't want to poo in a cup to help people?" And from one sense, that's all they're doing, but actually, they made contact, they came in, they spent their time, we pricked them with needles and took their blood, we scanned them, we put them through the ringer, and they did all that for nothing. So actually, that's an incredibly generous thing that they've done, and well worth pointing out. And we really whittled them down from 70 people who wanted to be donors. After we went through all of our criteria, did all of the things we did, only 10 would have been eligible, and we only chose 4 to be part of the trial. And one of those 4 is Sarah, who was also willing to talk to you today and tell you a little bit about why she was willing to be part of the trial.
Hi everyone, I'm Sarah, I'm 27 and I work in the city, and I guess I found out about this. I was I'm from south of Te Awamutu in the King Country, and I was home one weekend and was kind of scrolling through Facebook, and this ad comes up, the gut bugs trial, and I'm like, Mum's got a science background, and we sort of have always sort of been interested in gut bacteria and I guess the body's ability to absorb goodness from food and filter the bad stuff, but it was like, holy moly, this will be super exciting if this works. Like, I would love to be a part of it. And then so got in touch with Liggins and was definitely keen. And then you kind of come in and have all these tests and do all these funny things, and they know my body far better than I do. And I'm just kind of sorry to Ben and the team for having to deal with my staff donations. Yeah, I feel a bit bad about that. It's kind of weird. The first time you're like, oh gosh, this isn't normal. No, but it was like just a really exciting trial. Awesome if it kind of works. And yeah, I guess I was just stoked if I can help someone's health literally by donating some stool. I was super keen. Yeah, that's me.
Thank you very much to Ben, Alofa and Sarah. So as you heard, one member of the team who can't be here tonight is Tim Edney. Tim is a high country sheep farmer from Arrowtown and also a property investor. And he and his family recently made the decision to invest in the gut bugs trial also. I'm delighted that his wife Kath and daughter Rosalind can be here with us today. I think it's important to point out that the family made this decision very carefully and in fact in quite a scientific manner. They didn't just jump in, if you like. They thought about it carefully. They decided that this was research they really wanted to invest in, even if the outcome was uncertain. And in fact, Tim has said that the family don't much mind what the outcome is. It's really answering the question that interested them. And Tim has also taken a very in-depth role in the study. He attends the trial meetings and is integral to the study team. So the support of the Edney family really showed just how important philanthropic support can be in advancing the sort of cutting-edge research that we undertake at the Liggins Institute, that is helping us to answer key questions such as, can we turn off the obesity switch? So our heartfelt thanks to the Edney family. And if you would also like to consider supporting this or any other of the groundbreaking research of the Liggins Institute addressing key questions in health yourself, information on how you can do this is on the flyers that are distributed around the lecture theatre.